This is Duke University.
I'm Jerry Dawson, and I direct the Duke Institute for Brain Sciences. And now it's my honor to announce this year's winners of the poster awards. So in the area of cellular molecular mechanisms, the award goes to Danielle Clark, who's a graduate student in immunology, for her poster on the role of interferon gamma and STAT1 signaling in neuronal excitability and behavior. And then in the area of human research, the award goes to Jiahao Po, who is a postdoc in the Center for Cognitive Neuroscience, whose poster is titled Tuned to Learn, Anticipatory Hippocampal Pattern Stability During Mesolimbic Activation and How It Predicts Successful Memory Formation. So let's start with Danielle, who's going to give us a snapshot of her work.
So my study is looking at the role of interferon gamma, STAT1, in neuronal excitability and behavior. And we're particularly interested in looking at this in the context of autism spectrum disorder. So recent studies have shown that the immune system is actually playing a role in autism, and more specifically, the interferon pathway. So this is an antiviral immune signaling pathway. And we're seeing that it's one of the most enriched pathways in the brains of individuals with autism, which is really interesting. And so to see what a prolonged STAT1 activation in neurons may be doing in vivo, we've developed this mouse model, which uses a STAT1 gain-of-function mutation in which STAT1 can be phosphorylated normally, but prevents it from being dephosphorylated. And so these mice actually exhibit behavioral and cognitive deficits, as well as neuronal hyperexcitability. However, when we drive this mutation in microglia, the resident immune cells of the brain, these mice are normal, really suggesting that this is a neuron-specific role for STAT1. We still don't know how this prolonged STAT1 activation in neurons is causing these autism-like behaviors, but we're currently in the process of figuring out, filling in those gaps, finding those missing links, and hopefully getting a little bit closer to understanding how autism works and what causes it. Thank you.
So now I'm going to turn it over to Jiahao Po, and he is going to tell us how memory works and how we form them.
So we're constantly exposed to an immense amount of information with streams of new information coming from all our devices all the time. So depending on how curious or how invested you are actually in the event, you might remember everything that transpired, or you might just have a vague memory that it actually happened. So our motivational state in particular can play a critical role in determining what we learn and remember. So motivational influence on learning have been studied in humans using various forms of incentives that includes reward, extrinsic monetary reward, or intrinsic curiosity. So these studies have generally shown that increased co-activation in the reward-related mesolimbic brain circuitry and also in the hippocampus. But the nature of this interaction remains unclear. So here we investigate using fMRI how activation in the midbrain can modulate hippocampal state to influence subsequent learning. So over here, we adapted a trivia quiz paradigm that had been previously used to examine how curiosity influences learning. So prior to the experiment, participants went through a pre-screening where we selected a list of questions based on their subjective ratings, and questions were classified into high and low curiosity. So these questions were presented during fMRI scanning, and participants were shown the answer after a variable interval. So after the scan, participants received a surprise memory test where they had to recall the answer for the trivia questions. So unexpectedly, the recall answer was better for the high curiosity than low curiosity trivia question. So it does show that when participants are curious, they do tend to learn the information better. And over here, if we look at fMRI activation, we see that in the VTA, so the midbrain VTA, there is higher activation in the VTA when participants are anticipating high curiosity answers. And that moves us on to our primary question. So how does this increased activation in the VTA influence hippocampal states? So to look at hippocampal states, we came out with a measure that we call the analysis of hippocampal stability. And what we did is to look at when questions are presented, how does the average pattern, so the centroid of this activation pattern, look like? And we can then quantify the activation pattern relative to the centroid. So how far is this activation pattern from the centroid? So if it is closer, we consider it to be more similar to the average pattern and thus more stable. So looking at the results over here, so if you look at the y-axis on this, so it represents a representation distance with shorter distance representing items that are closer to the centroid. and thus more stable. And we see that when participants are curious, the hippocampus seems to be more stable. It exhibits a higher stability. And most importantly, when these patterns are more stable, they are also more likely to learn the answers. They're more likely to recall the associated answer. Finally, looking at VTA, and we see a clear relationship over here as well, which is that when there's greater VTA activation during anticipation, they also show a greater hippocampal stability. We showed that trivial questions that elicit high curiosity biases distributed patterns in the hippocampus towards greater stability. And this increase in stability was also associated with greater recall of the anticipated trivial answers. And most importantly, the stability in the hippocampus was strongly associated with trial-by-trial variation in anticipatory VTA activation. And based on that, we propose that engagement of the dopaminergic midbrain supports motivated learning by creating a stable learning state in the hippocampus that supports the formation of new memory. Thank you so much.